If I start really from the reason, the purpose why we are interested in this, that the discussion in today's society is very threat focused, so that we're talking about climate change and resource depletion. Now it's the pandemic time, so there are of course a lot of crises that are threatening humankind, but it's also at the same time that we have perhaps the best tools ever at our use then to combat these challenges and turn these things into things that with humankind can thrive. So science and technology can provide hope during these types of times, but it's also something that we need to choose to support now and really invest into that area. And VTT exists for this sole purpose. Our focus is on finding solutions to these grand challenges through science and technology. The impact that we create comes from our work in science and technology, but it only acquires the value and the impact when a company applies it in their products and services. It can be also, of course, a part of society, that kind of like public authorities that utilize these technologies. And in everything that we do, we want to be doing things that really go a little bit beyond the obvious, so that we apply science in a new way, or we can find a solution to something which previously has been really difficult. And when you're talking about beyond the obvious, synthetic biology is smack in the middle of that definition. The fact that we can now start utilizing modern understanding about biology and biotechnology and these types of things, but also how we can apply digital technologies in novel ways. So it's also kind of like things like robots or deep learning. In the future, it will be quantum technologies, quantum computing through quantum biology that we can apply there. And we can actually modify these organisms in a way that they can produce something that has desired properties. We can utilize it then to really bring solutions to market that were impossible in the future. I think that that's astonishing, but it's also very necessary then if we think about the challenges that we then face in creating renewable materials, creating solutions that are climate friendly and promote all the right things. We also kind of like have seen that how fast this field has been developing in the past years, and one example of this is of course like the Millennium Technology Prize in 2021 that was awarded by the Finnish president to Subramaniam and Kleinerman for their work on gene sequencing. That has been one of the enabling technologies there that has made it also possible to make such leaps forward in synthetic biology. Now, from VTT's point of view, this of course comes then to the research that we are doing. That our job is to help in a way speed up and scale up the impact of this. So through the research that we're doing, through the people that we have, through the infrastructure, research and technology infrastructure that we have, we can help companies then utilize this knowledge that there exists, and without having to invest too much in the beginning, help start learning about these technologies, but also then that VTT can help then speed up the time to market from idea to innovation, but also then that we can scale up from laboratory scale then to the real factory scale, so that it can become commercial and really be both a commercial success, but also then bring these things to fruition. And this, of course, all the work that we do, nothing happens alone. So that nothing happens by only of course by our people. That this is a very collaborative field. It's very much happening in networks or ecosystems, and like in nature, it is very important that the ecosystems are as diverse as possible, so that we can bring different type of skill sets to work together, but also then that we then share the same goal, that we are working towards the same purpose but then applying the different skill sets, and research organizations can have a huge impact also by facilitating this and making it possible then for large and small companies, universities, NGOs, whomever, to be working together and creating then those kinds of thriving ecosystems. And we are, of course, very involved in a lot of EU activities and active in the networks that exist in the synthetic biology area there. But as a whole, for VTT, synthetic biology is one of these areas where we believe that we will be creating absolutely gigantic impact for the good. It is one of these technologies that are breakthrough technologies. Some of the digital technologies are perhaps a bit better known in the general public's eye, but I think that we will know that how much good will be created through synthetic biology and investing into research and innovation work there.
Thank you, Antti, for the inspiring start. Next, we will hear from the Synbio Powerhouse co-creation manager, Tuula Palmén. Please.
Welcome also on my behalf to this annual Synbio Powerhouse event. The more active promotion and development of Synbio Powerhouse was started about 3 years ago. So this is a kind of anniversary event, 3 years of Synbio Powerhouse. The goals of Synbio Powerhouse is to increase and accelerate the use of synthetic biology, advanced biotechnology in Finland and beyond. In these 3 first years, we have developed services that support the pathway from research to business. And especially we have been focusing how to encourage people to innovate and then help the innovator teams to turn the idea into product. And the other way we have supported is that we have been made it easy to access to ecosystem. And one of the goals has been that we co-create joint projects, and that has been really interesting and successful. Once again, welcome to our event. Enjoy the program and innovate.
Our next speaker is the founder of Synbio Powerhouse and one of the world's foremost synthetic biology scientists, Professor Merja Penttilä.
Welcome everybody on my behalf as well. We are living exciting times because sciences are developing so rapidly, and it is no doubt that their integration, the interdisciplinarity, would bring great innovation potential and disruptive added value. On the other hand, we are living challenging times. As we all know, we need to combat climate change and to find ways to make a transition from fossil oil to sustainable solutions, both from the raw material point of view and the product point of view, to make the green transition in manufacturing possible. We believe that biotechnology should have and will have much more to offer for our future. Envisioning that we can use the synthesis power of living systems, such as microbes, in closed industrial production plants to manufacture basically everything we make today from fossil oil sounds like a radical thought. And the technology is in many ways radically different to all the other manufacturing technologies we have. This is why it may also be difficult to understand for those not in the field what all biotechnology could offer. One may get lost with all the gene acronyms, the gene name acronyms, and on the other hand, it is its enabling nature may not be as clear that one can produce products for various applications for fuels, pharma, materials, chemical industry, and so on, and that this can be done basically just engineering the code, the DNA, that then dictates what the cells can produce. Biotechnology has many benefits. So one of them, and an important one, is that it follows intrinsically the rules of circular bioeconomy. Various complex raw materials and waste fractions can be used as feedstock for the microbes, and the products and process streams are recyclable and reusable. With the new tools of synthetic biology, we can make strain engineering much more rapid and predictable, and aim to make biology much more engineerable, and thus overcome the fact that biology is complex. We are actually on our way to biointelligent manufacturing, which is expected to become more and more important after 2030. This is how we see it at VTT. We can utilize the vast information of genomes and the biological functionalities, and in particular with the use of AI, machine learning tools, to make sense out of this information and pick up the best genes, for instance, for our purpose. A third pillar here on the left is the concepts of circular bioeconomy. We can then use this information to design production organisms through the so-called design, build, test, learn cycle of synthetic biology to obtain, in an iterative manner, superior strains for the real manufacturing process. We aim for a large number of examples where biotechnology can deliver sustainable solutions and processes that can be automatically controlled. We aim to fulfill the promise that we can demonstrate a large product range, even new to nature chemicals and materials, and design the bioprocesses to be carbon neutral. We aim for AI-assisted cell and process design and autonomous process operations. Finland has been already very early in the 80s, one could say, applying gene technology in industrial biotechnology. For instance, in engineering brewer's yeast to avoid formation of buttery flavor in lager beer, or in understanding how the amazing atomic level structure of wood hydrolyzing enzymes, cellulases, aid in biomass decomposition. This seminar is focusing on examples more of today's biotechnology and synthetic biology in Finland, both at the industry and in the research labs. This way we hope to increase the understanding of biotechnology and synthetic biology and get you all inspired of their potential and importance for the future.
Thank you so much, Merja. Aligned with the bioeconomy theme and cross-disciplinary collaboration, our next speaker is Professor Orlando Rojas, who is the Scientific Director of the Bioproducts Institute at the University of British Columbia. And here at Aalto University, he's the Principal Investigator of the Finseres flagship program. Orlando, what inspires you in this time as new collaboration and multidisciplinary action is required to mitigate the climate crisis and the resource deficiency?
So my thinking is that while the challenges associated to our energy and material needs have become global, most of the solutions are taking place in a more granular manner, that is locally. For instance, to the space and geography, we could perhaps add time, since the sense of urgency, for instance, with respect to climate change, It's different depending on the location where we live. So science-based efforts that are being deployed involve quite numerous cross-cutting disciplines across the globe. And this includes those that relate to environmental sciences, chemical, physical, biological, social, and economic aspects. So divergence and convergence of the science is not an oxymoron. Now, more than ever, we need a specialization, and at the same time, the boundaries between disciplines should become more blurred, given also the tendency that we see nowadays in university systems and research centers to work on project-based models. So this interaction between researchers in an ecosystem really should lead to better learning and innovation. And in that sense, institutions around the world need to work together and wrap their heads around real-world problems. And this, for instance, requires students and researchers from different areas to work together across the globe, given that in different places in the world, the challenges are tackled in different manners, and perhaps there are many complementarities. So this is very important and calls for an action that should be global rather than local.
How do you see the opportunities for the forest industry to valorize some of the side stream sugars using biotechnology?
So my thinking here is that sugar streams produced from fiber processing are really currently at least underutilized. And here the power of biotechnology cannot be underestimated. I have learned, for instance, recently about the hybridization of typical pulp mills that not only produce wood pulp, but also are able to use residual sugars as a culture medium with microorganisms. An example could be, for instance, the production of bacterial cellulose. In the same manner, we can say similarly about efforts to use fermentation processes to synthesize polymers. And in this sense, one of the main attractive elements is here the possibility to produce molecules, oligomeric or polymeric compounds that are more uniform in their profiles. In such a way, we need to use feedstocks that can come from streams, from production lines, or from the end of life of recovered materials. And again, the possibilities here are enormous. And this really provides us a beautiful example of the regenerative nature of lignocellulose resources, which are not only sustainable and renewable, but can be inserted in a truly circular ecosystem.
Last, I would like to ask how the power of biocatalysis is in fact present at the Bioproducts Institute currently.
I can speak about our own efforts in the Bioproducts Institute here at the University of British Columbia. So one of the 5 pillars that we have in the Bioproducts Institute is biocatalytic transformation and engineering. And that signals the importance of this area in the context of the lignocellulose processing. The theme encompasses the biosynthesis and biological degradation of biomass, as well as the engineering of related processes, for example, to extract and valorize different biomass components. In this sense, we're carrying out fundamental research to understand how genes and their products function in the context of complex biological systems and synthesis or assemblies, or also in the degradation of biomass. So in this area, applied research is being deployed to look into protein engineering and synthetic biology to design biocrops and biocatalysts. The applications here, for instance, include the development of new biocatalytic routes to valorize biomass, or, for instance, to reduce the energy requirements in material production, or the functionalization of biomass by novel routes used in biology, and with an impact, of course, in materials manufacturing.
Our efforts to significantly decrease the use of fossil raw material puts high pressure on developing sustainable alternatives. And this means putting to use all the potential side streams, byproducts or waste streams that are generated from renewable sources. Everything that is created in nature has a counterpart that can degrade and recycle those carbon-rich molecules. Here, biotechnology offers immense opportunities. Using biotechnology, we can combine in novel ways activities found in nature to consume pretty much any organic compound found on this planet. Or at the same time, we can create new compounds and new materials.
In nature itself, it depends on the organisms, what their capabilities are in dealing with the waste. If we have molds and mushrooms, so they are very good at utilizing heterogeneous organic material, like sawdust is there, different plant materials. If then we have yeasts, they very much like sugar, so we know that you can produce ethanol from sugar with yeasts. And they are not so good at very complex materials like the fungi. Then, which is beautiful, also biological organisms can utilize simple carbon sources as CO2 and convert them further. And in addition to sunlight, you can also use hydrogen to convert CO2 in the metabolism of the organisms. So from these raw materials, which are waste fractions, you can then build in the metabolism of the cells very elaborate and complex and very functional components which form the cells of different types of chemistries, from carbohydrate, lipids, proteins, pigments, molecular motors, sensor molecules, and so on. So the capabilities are enormous.
And all this is, of course, catalyzed by enzymes that the cells create. Ideally, we need to use this power to turn all our waste streams into materials that need a carbon source, instead of taking that carbon from fossil raw materials. And this is how we see the future biorefineries, a combination of processes all optimized for the maximum utilization of natural, recyclable, or non-fossil carbon sources. Today we're going to look at many examples of this in various applications and various stages of research, from fundamental findings to demonstration or pilot scale-ups. Our first case example comes from St1, the Finnish bioethanol producing company, who took a bold step forward to produce their own enzymes needed in the bioethanol production from lignocellulosic waste streams.
Bioprocesses have actually been familiar to us for 15 years, since we have been an ethanol producer for a long time from bio-waste. So in that sense, producing valuable chemicals with bioprocesses was a familiar thought to us. And we figured that enzyme production is really not that different from ethanol production. So you take sugars, you take a carbon source, you take a nitrogen source, and then you get the product that you want. So in that sense, including yet another bioprocess to our biorefinery seemed to be quite a natural thought.
Producing your own enzymes has clear benefits.
It's mostly to do with the price of enzymes. So it is one of the top 3 costs when producing 2G ethanol out of softwood biomass.
The plans are to include the on-site enzyme production in a future commercial-scale St1 Cellunolix biorefinery.
We have the Cellunolix process, which first has the pre-treatment of the softwood sawdust, and after that we have enzymatic hydrolysis, and after that we do a fermentation and distill the ethanol from the process. In addition to ethanol, we also have other bioproducts from the refinery, for example, an organic grade fertilizer, and also lignin, which is currently burned, but a lot of research is carried out in order to valorize it further. And into this biorefinery in the future, we're hoping to include an enzyme production unit, which will preferably utilize some of the streams from the main process as carbon source and produce the enzymes to be used in the hydrolysis step.
If you make your own enzyme, you can reduce costs involved in transport, in stabilization of the commercial enzyme products. You are not dependent on third parties, let's say, in your process development. But there is also like a more technical part in here. So the biomasses and the pretreatment conditions produce very different kind of materials, and there are only like limited amount of commercial products available for this purpose. And if you think the chemistry of the biomasses that should be depolymerized by the enzymes basically in that process can vary very much between different biomasses and also different processes. So the one solution is not okay for every purpose. And for that reason, that was the second reason why they wanted to have an own enzyme product, which would be optimized for their system.
But why use enzymes and not acids to treat the lignocellulose?
Enzymes are biological catalysts which are very specific. They can operate in mild conditions, which is very nice. They don't produce any side products because they are specific, compared to, for example, acid hydrolysis. In this enzymatic hydrolysis step, the requirement is that you convert as much of the cellulose and hemicellulose into monosaccharides as possible, so that the yield of the hydrolysis process is high, and you don't. There is no residual polysaccharides basically after the hydrolysis.
What was the main challenge in the polymerization of the lignocellulose?
There is a need to have many different kinds of enzymes, and the composition of the enzymes optimal for different biomasses is different in each case. And for that reason, there is a need to optimize the enzyme mixture for different biomasses and pretreatment conditions, because the substrate of the enzymes is different in each case. And for the enzyme mixture optimization, we usually do so that we first take a screen from VTT's collection and the enzyme mixtures produced by the VTT's fungal strains, and test these mixtures in the hydrolysis first, in conditions mimicking the process as close as possible. And from these mixtures, we select the best ones for the continuation. After that, what we do is that we add in this baseline enzyme mixtures new enzyme components, which can, for example, hydrolyze oligosaccharides or attack on the crystalline cellulose or attack on the more amorphous parts of the cellulose, or attack on hemicellulose, depending on what kind of biomass is there in process design. And by these kind of debottlenecking experiments, we identify that what enzyme components we need to add in this baseline enzyme mixture so that the hydrolysis yield will improve.
I'd like to ask Merja, how can synthetic biology be applied in this type of enzyme research?
So first of all, we can for instance, change the properties of the enzymes. So we can use so-called protein engineering, where we can alter the amino acid structure, and thus we can improve, for instance, the pH tolerance or the temperature tolerance. We can also put up sort of directed evolution, where we have had the Nobel Prize winner, Francis Arnold, and the Millennium Prize winner here in Finland to obtain the prize for. So this is a field which has been there for a long time. Then another thing which is important, in particular for this ST1 project as well, is that when one is looking for new enzymes, just taking the genes and then putting them into the production organisms like trichoderma. So the problem is that trichoderma naturally produce a huge amount of enzymes, which activity impairs or is problematic if you want to analyze the activity of a new enzyme candidate. And now then, with synthetic biology, we have been able to design synthetic promoters which drive expression of the enzymes in conditions where the other enzymes, the natural enzymes, are not expressed. And thus you get a pure enzyme. And you can also do this in a high throughput manner, so you throw the genes in under this synthetic promoter, and then you take the fungus then secretes the enzymes, and then you can measure the activity individually of a large number of fungal clones each producing a specific new novel enzyme.
Typically, the starting point of the in this enzyme mixture development, the hydrolysis yields can be around 60% or 50%, 60 to 50%. But by this optimization process, we usually can achieve tens of percent improvement in the hydrolysis yield. For this debottlenecking or screening of needed enzyme activities, we use VTT's own enzyme collection. We have a range of different kind of hydrolytic and oxidative enzymes which can improve the hydrolysis or overall depolymerization of the cell wall, the lignocellulose polysaccharides. But we are not only relying on the existing enzymes. So, for example, in this ST1 case, we have utilized the enormous amount of sequencing data, fungal genome sequencing data that is available in the world. So that has opened up a totally new type of insight into the genomes of fungi and the genetic diversity of the enzymes that act on this lignocellulose hydrolysis or depolymerization. We also at VTT have our own databases. We are not relying only on the public ones. These are generated in such way that we cultivate the different types of fungi on different type of lignocelluloses, and when these fungi grow in this kind of lignocellulose containing media, they start to produce or express genes which they need to be able to grow in these conditions. And after that, we isolate the RNA from these fungi and sequence it. And then we can find out what genes the fungi actually has expressed in these conditions and what enzymes these genes encode. And in that way, we can select from these sort of transcriptomes the enzyme genes which are good candidates for the improvement of hydrolysis of that kind of lignocellulose. And these genes are then transferred to this industrial production host. So if you want to produce enzymes industrially, you have to bring the natural genes into an industrial production host. Otherwise, they are not available for any process. At VTT, we typically use this Trichroma resei strains, which have a capacity of producing like 50 to 100 grams per liter of protein in bioreactor conditions. How we usually do it, for example, in this ST1 case, we had the good baseline starting strain. We put the new genes out from the which were found in the genome screening approach, and make a new engineered trichoderma strains which have these new enzymes, and in that way improve the enzyme mixture that the fungus produces.
The genetically modified fungal strain has superior hydrolytic capabilities.
The new enzymes what we introduced in this system, so typically by adding 2 to 4 enzymes, new enzyme genes, you can improve, by good selection you can improve the hydrolysis yield like to 90% depending on the biomass type.
In nature, trees would remain for thousands of years if it weren't for the degrading fungi and the numerous enzymes they produce.
Lignocellulose is extremely complex, and I think nobody even now knows what all enzymes are involved in the degradation in the nature. And if you think about how many different kinds of microbes are involved in biomass degradation in nature, so the range is enormous. Even within one enzyme class, like I have a xylanase which hydrolyzes linkages in xylan backbone. So even in that class there are like very many different types of variants. Some operate which have different properties, or the catalytic activity is basically the same, but they can have different pH range, temperature range, other properties, which are important to understand in this industrial enzymatic hydrolysis. About these new enzymes, I think one of the significant findings in, well, relatively near future was discovery of these cellulose oxidizing or polysaccharide oxidizing enzymes, LPMOs, lytic polysaccharide monooxygenases, which had this remarkable ability to increase the hydrolysis when they are introduced into a mixture containing common hydrolytic cellulolytic enzymes. So I think that is a good example how even in this quite modern days we have found new enzymes which are extremely significant in this lignocellulose hydrolysis.
The oxidizing enzymes originally discovered by Professor Vincent Eysinck are currently a hot topic in enzyme research. But how do they differ from cellulases?
So what LPMOs do, they attack in the cellulose polymer, and they can attack on the crystalline parts, so that they create like defects in the cellulose crystals, and so that the other enzymes can access those better. And the difference between LPMOs and cellulases is that there are cellulases which attack on crystalline cellulose, but they start from the chain ends. So they don't cut the crystals inside. There are enzymes which are able to hydrolyze cellulose within the cellulose chains, but those are not active on cellulose crystals. So these are specific enzymes for creating nicks inside the crystals, so that for example, other enzymes called cellobiohydrolysis can start hydrolyzing the crystals more efficiently. And the cellulose crystals are the most recalcitrant parts of the cellulose or polysaccharides within the lignocellulose, basically.
The future St1 biorefinery might even have different main products.
I find it quite possible that in the future we have a biorefinery that produces ethanol, but the main product is actually something completely different. If you look at the oil distillation kind of curve and the stuff that you get from the oil refining process, there are a lot of other components to be replaced with non-fossil products as well. So it's not only the liquid fuels, it's also the different kind of polymeric materials and stuff like that you are able to produce while producing, for example, ethanol. So in that sense, if we get a great value from for example, our lignin, I don't see it as impossible that we have at some point a lignin refinery that produces ethanol as well.
The pulp and paper industry could potentially offer many opportunities for synthetic biology. Current pulping methods create side streams that are mainly burnt in the mill. However, exploring how these could be turned into high-value products is of increasing interest. Today we will be looking at several examples of using cellulose, hemicellulose and lignin to feed microbes for the conversion of various end uses. The largest underutilized side stream of pulping is hemicellulose. Around 15% of the dry content of wood is made from these C5 and C6 sugars. These sugars can be fed to microbes to produce plastic precursors, food additives and various chemicals. In a previously published study, Merja Penttilä's group was able to turn the black C5 slurry into pure xylitol using modified yeast strains. You can watch the whole story of exactly how the strains were engineered on our YouTube channel after this seminar. We featured this story earlier this year in the VTT webinar on synthetic biology. In this study, the hemicellulose was from a fairly rare process. However, millions of tons of extracted hemicellulose side streams are burnt annually. And these side streams are the pre-hydrolysis of dissolving pulp mills. To understand more about this side stream and the biotechnological potential it bears, we've asked Pat Cavanagh to give insight into the dissolving pulp market and what the global volumes are like. Pat is an economist at RISI Fastmarkets, the leading pulp and paper market analysis company. They have very recently published an enormous 200-page report on basically everything there is to know about the dissolving pulp markets. So if anyone knows what's happening in this field, it's definitely Pat Cavanagh. So Pat, what is dissolving pulp mainly made for?
The largest end-use demand segment for the dissolving pulp industry is textile fibers. Cellulosic fibers have been the main engines of growth for dissolving pulp consumption over the past 10 years.
And how does the dissolving pulp market look now and in the upcoming years?
Dissolving pulp consumption grew at an average annual rate of nearly 7% over the past decade and reached over 8 million tons in 2019. We forecast that demand growth in the decade to come could easily outpace even the rates exhibited in the last 10 years. This places dissolving pulp easily among the fastest growing segments of the forest products industry, with significant upside potential considering the sheer size of the global textile fiber market at over 100 million tons per year.
What does this mean if we think about the volume of the hemicellulose side streams?
With respect to the annual potential volume of pre-hydrolysis hemicellulose side streams, it's important to first say that dissolving pulp demand is met from a range of sources. These include dissolving wood pulp, cotton linter pulp, bamboo, and modified paper-grade pulp. If we focus only on the dissolving wood pulp portion of the market, total capacity in 2021 is estimated to be about 9 million air-dry metric tons. Of that, 1/4 is softwood-based and the remainder is hardwood-based. If that capacity were to run fully, it would consume about 23 million tons of wood fiber. If we then apply the average hemicellulose content for hardwood and softwood species, we arrive at an estimate of about 6.5 million tons of potential pre-hydrolysis hemicellulose side streams. Of course, a range of factors can influence that estimate, including actual operating rates at dissolving pulp mills, market conditions for softwood and hardwood grades of pulp, and the precise species and hemicellulose content of the wood fiber consumed at each mill. The dissolving pulp study helps to unpack this problem by providing precise operating rate and production forecasts based on the performance of end-use demand markets, along with capacity forecasts by specific grade and process of dissolving pulp.
Thank you so much, Pat, for this valuable insight. And for all of you out there interested in this massive report, you can purchase it from the Fastmarkets RISI webpage. Bulk-grade cellulose is mainly produced by kraft pulping today. In this process, there is no pre-hydrolysis. Professor Olli Dahl from Aalto University proposes another method to extract the hemicelluloses, that is, in post-hydrolysis. This process would offer many high-value products and clean sugars not containing any harmful substances.
My vision is that we do not use pre-hydrolysis. Instead of that, we use post-hydrolysis. The kraft process itself removes all those nasty compounds, fat and resins, acids, turpentines. It removes them first. And finally, you have the pulp, which do not contain those things at all. If you don't use pre-hydrolysis, you can maximize the hemicellulose content in your pulp. And then the hemicellulose is very pure.
Olli and his associates hold the patent for AltoCell MCC.
When you make MCC using, for example, AltoCell method, you will get very nice streams of pure sugars. And these sugars are so pure that you can use them as such wherever you want to use it.
This compound is an interesting friend to microbially produced polymers. For example, producing PLA and MCC in the same mill would make a lot of sense, since they make a wonderful biodegradable composite together. For us at Synbio Powerhouse, new ways to collaborate with the forest industry is of highest interest, combining the solid know-how and long tradition of pulping in Finland with new biotechnological pathways of utilizing the side streams. In a recently established Business Finland project, precisely this angle is being explored. The project is called Synbio Pro, and here researchers at VTT and Tampere University are working on turning agricultural and forestry residues into platform chemicals which could replace fossil-based precursors for plastics. The project aims to create solutions throughout the value chain in collaboration with industrial partners Neste, Kemira, Fortum, Olfactomix and Merja. Neste is the world's leading producer of renewable diesel and sustainable aviation fuel. Neste is also focused on developing renewable solutions to the plastics and polymer applications. Currently, the company is scanning for new ways to replace crude oil.
First of all, we want to be focusing on raw materials that are of course sustainable but also globally abundant and scalable. So we have been looking at the potentials of different kinds of waste and residue raw materials and see that, for example, lignocellulosic residues like agricultural residues such as straw or forest residues, they have the global potential of billions of tons annually. Likewise, the municipal solid waste is produced in the billion-ton scale annually. Also, very important in the future, there will be higher availability of renewable electricity, which also allows to develop new PTX type of products that are produced using renewable electricity and that are not dependent on the biomass availability.
What role does Neste see in biotechnology and synthetic biology?
We see industrial biotechnology and synthetic biology as a promising technology platform for these solutions. We are developing new technologies and businesses that can utilize scalable and abundant waste and residue raw materials, or even carbon dioxide via microalgae or Power-to-X. Synthetic biology offers possibilities to convert these materials into sustainable platform chemicals, polymers or fuels more efficiently than before, with higher carbon yields, less side products, and increased energy efficiency. Synbio Pro project is a promising synthetic biology initiative where we are collaborating with VTT, Tampere University, and companies along the value chain to develop more efficient pathways to produce important platform chemicals like acrylic or adipic acids from renewable raw materials, namely lignocellulosic sugars or lignin. These platform chemicals are used in polymers that are utilized in everyday life items like clothing, diapers, carpets, or vehicle parts. We in Neste have the know-how and solid track record to develop, engineer, and scale up new sustainable technologies into global scale businesses. We collaborate with research institutes, universities, technology startups, and value chain partners in developing and commercializing renewable and circular solutions that help to mitigate climate change.
In the Synbio Pro project, two platform chemicals, acrylic acid and adipic acid, has been selected. Why these acids, Perttu?
Both acrylic and adipic acid are produced in the scale of millions of tons globally annually and currently from fossil resources. So by having a drop-in solution of replacing the fossil counterpart with the renewable counterpart, there is market demand and easier access to the market.
For Kemira, one of the world's leading specialists in pulp and paper and water treatment chemistries, Synbio Pro offers a window to new innovation opportunities.
We've made this strategic commitment now to sustainability. It's high up in our strategy. We are aiming for profitable and sustainable growth. And we have 5 focus areas there. So we're talking about safety, we're talking about people, we're talking about circularity, water, and climate. And especially if we're thinking from a like an innovative product point of view, in that circularity becomes very, very important. And of course, we see the pressure also from customers. So our customers require sustainable solutions from us. And from that point of view, again, that's why we have focused to focus on bio-based, to say. So in the bio-based context, we have a concrete target. We're heading for 500 million in bio-based revenue by 2030. And in that regards, both synthetic biology and biotechnology are, of course, interesting solutions to take those targets forward.
The research is divided into two main projects: the utilization of lignin for the production of adipic acid, which is conducted by Ville Santala's lab at Tampere University.
So if you think about the traditional industrial biotechnology, usually the strange microbes that has been used are easy to engineer, so genetically engineered, and for example E. coli and yeast, and they are very good to convert carbohydrates, for example glucose, to the product in the industrial relevant metrics, so to get to good rates and yield and titer. But if you think about, for example, circular point of view and some industrially produced side stream that doesn't contain carbohydrates, instead they can contain organic alcohols, acids, aldehydes and aromatics that can be even toxic for those traditional industrial workhorses. So how to get the carbon from those streams? So one approach is that we take the microbe that can utilize those compounds, and then we use that as an engineering platform. It's common thinking that it's much more easier to construct the production pathway to organism than to integrate the catabolic needed, for example, to utilize lignin-derived molecules as an example of this kind of aromatic compound that are toxic for the common hosts.
Tampere University has specialized in an exotic strain of bacteria called Acinetobacter bailey ADP1. As one of the few labs in the world focused on this bacteria, the Tampere team has been able to unlock some of the useful potential this organism has to offer.
The best benefit is that it can naturally consume the aromatic compounds that are derived from lignin. It has the catabolic roots for that, which E. coli does not have. It is a versatile bacteria. It can consume a broad range of substrates such as decarboxylic acids, carboxylic acids and aromatic acids, and of course sugars. And it also has rapid doubling times, and it can grow on very limited media. So for example, just using a minimal salt media with one of the aromatic substrates as a carbon source, it can grow quite nicely. And it also has this high tolerance towards inhibiting compounds. So if you think about lignin hydrolysates, they are a complex mixture of different aromatics, but also, for example, acetate and residual sugars and other impurities. And many of the compounds found there can be inhibiting in low concentrations for many common host organisms. But our host organism, it can quite readily consume the substrates and also tolerate the compounds.
And what about the engineering of this exotic host organism?
In addition, for our host organism to have these beneficial traits for bioindustrial processing, it also has very nice traits for genetic engineering. So it is naturally competent, and it uses homologous recombination. So it kind of gives flexibility on designing the genetic insertions for the host organism. And even though it's a quite new host organism, its genome has already been experimentally annotated, and it has curated metabolic models. And also these genetic engineering tools are already available for it. So kind of combining these good properties as a bioprocessing host organism for lignin-derived molecules, it also has the ease of the genome engineering for us to engineer this organism as a cell factory to produce industrially relevant and interesting compounds.
At VTT, the focus is to utilize the sugars in lignocellulose using yeasts.
So at VTT, we aim to produce acrylic acid from lignocellulosic sugars. So many acids are used as raw materials for plastic production. Acrylic acid is used as a platform chemical for water treatment and for diapers. So it's a super absorbent polymer. Currently, acrylic acid is produced through oxidation of propylene and propene. So we are here now looking for more sustainable production options. So we aim to replace the fossil-based raw materials by lignocellulosic sugars. Side streams from forest industry and also agro waste. At VTT we have a large strain collection, so there are different microbes, and that allows us to pick the most suitable production hosts for each project. So for this project it's of course important that the host is acid tolerant, as we plan to produce acid. So we have here selected an acid tolerant yeast, and VTT has a long history of producing organic acids with yeast. So we have earlier produced lactic acid and muconic acid, for example, and we can of course use all that expertise also in this project.
As a starting point, are there any natural organisms that produce acrylic acid?
There are no known organisms that would produce significant amounts of acrylic acid. We need to engineer the metabolism of the yeast. We need to introduce new genes from other organisms into our yeast. And in addition, we can block some non-essential pathways so that we then direct the carbon flux more efficiently towards acrylic acid. There are some known pathways for acrylic acid production that have been studied already by others, and we have been modeling those. There is, for example, one going through malonyl-CoA, and then there is another one going through beta-alanine, and we have been modeling those and also some other options, and we have then picked the most promising ones, and now we are introducing those into our yeast host.
The Finnish energy company Fortum is also part of the Synbio project. The company is interested in diversifying the long-term application potential of the hemicellulose and lignin fractions derived from lignocellulosic side streams. Of particular interest is the agricultural straws, which will be the feedstock in Fortum's future biorefineries. Fortum sees that collaboration between research institutes, biorefinery developers, and chemical and material industry is required to accelerate formation of hemicellulose and lignin-based value chains, and eventually the commercialization of new bioproducts.
The industry collaboration is very valuable in this project, so it's very important for researchers to get the feedback from industry, so we then know that we are really going in the right direction.
Another abundant feedstock readily available for biotechnological utilization are the food industry side streams. Microbes are naturally adapted to feed on carbohydrates that are inedible for humans. Our next case example will take us from a bold idea to lab-scale concepting and finally scaling up and piloting. In this fascinating case, a Finnish insulation company called Finfoam developed a bio-based alternative to the fossil-based polystyrene insulation material using a food industry side stream as feedstock for the microbes. The CEO of Finfoam, Henri Nieminen, how did it all begin?
No se kaikki lähti siitä, että Uudessakaupungissa tämmöinen soijatehdas meni konkurssiin ja se oli hyvin pitkäänkin siinä, että se ei kelvannut kellekään. Ja me ajateltiin, että kyllä tätä on sen verran kuitenkin moderni ja uudenlainen laitos, että kyllä se nyt pitäisi pystyä jotain tuottavaa työtä tekemään ja tartuttiin siihen haasteeseen ja varsin heti niin kuin huomattiin, että nämä kaikki sivuvirrat, millä ei ole juurikaan niin kuin varsinaisesti arvoa, niin meidän pitäisi niin kuin niiden arvoa pystyä kasvattamaan, jotta tämä laitos pystyisi olemaan niin kuin kokonaisuudessaan kannattava. siitä se sitten lähti oikeastaan, että ruvettiin tutkimaan tätä soijamelassia, että mitä me voitaisiin siitä tehdä jotain järkevää. Myös oli soijan kuori, sitä on myös jonkun verran tutkittu, mutta tämä soijamelassi oli niin kuin kuitenkin se ehkä sit mielenkiintoisin, koska siinä alussa oli se tilanne, että me jouduttiin jopa maksamaan siitä, että me päästiin siitä eroon. Määrät on valtavia, jolloin tietenkin se on jotain mahdollisuutta kehittääkin, koska silloin, jos sulla on pieniä määriä, niin ei siitä niin kuin juurikaan sit saa mitään irti, mutta kun sulla on heti siinä putken päässä niin iso määrä tämmöistä biomassaa, niin siitä pystyy jotain ehkä onnistua tekemäänkin.
The research project began with the characterization of the soy molasses.
First of all, we analyzed the composition of the soy molasses, and we also experimentally tested if microbes can grow in it. And yes, microbes can grow in it. It contains lots of carbohydrates, so mainly sugars. For example, sucrose, which is the table sugar, but also oligosaccharides, which are quite special. So it's like galactose units linked to sucrose. There can be from 1 to 4 galactose units attached to sucrose. And these oligosaccharides are such that the yeast cannot naturally utilize.
The cell engineering challenge was to engineer the yeast strain for the digestion of the oligosaccharides.
There are different alpha-galactosidase encoding genes known in the literature, so many fungal species can produce alpha-galactosidase, which can cleave the galactose units from the oligosaccharides, and yeast itself produces the invertase, which can cleave sucrose. The solution to enable the yeast to utilize the oligosaccharides was to transform an alpha-galactosidase encoding gene from a different fungal species into the yeast. The transformed yeast would then synthesize the invertase and alpha-galactosidase enzyme inside the cells, and the enzymes would be secreted out of the cells into the culture medium, where they will attack the oligosaccharides, and they can then cleave the oligosaccharides into the monosaccharides, which the yeast can then metabolize further in the central carbon metabolism. And then the next challenge was to make the yeast produce lots of lactic acid, which is something that the yeast does not normally do. But yeast have been previously engineered for production of lactic acid, and it can be achieved by introducing into the yeast lactate dehydrogenase encoding gene from lactobacilli. And at VTT we have a long history of engineering yeasts for the production of lactic acid and also for the production of other acids. And we have isolated different yeast species or strains which are especially tolerant to higher acid concentrations than normal, like random baker's yeast strains. And this is an important feature when the product is acidic and can cause severe stress to the cell unless it can cope with the acidity. So we then combined the different features required for soy molasses fermentation into lactic acid into the same yeast cell line. So the production of oligosaccharide hydrolyzing enzymes that are secreted and the ability to produce lactic acid, and also we decreased the ability of yeast to produce alcohols because yeast metabolism is optimized for the production of ethanol, and because ethanol and lactic acid are made from the same precursors, they are competing reactions, and in order to be able to produce large amounts of lactic acid, ethanol production has to be decreased to a low level. And this can be done by knocking out the main genes responsible for ethanol formation, which are pyruvate decarboxylase and alcohol dehydrogenase. Then, when the engineered yeast was grown in soy molasses medium, we observed that yes, it can produce lactic acid from the soy molasses, and at the same time we saw that the oligosaccharides were disappearing from the medium as they were degraded and the cell was taking them up. We also tested a few different alpha-galactosidase enzymes in order to see whether some of them were more efficient in oligosaccharide hydrolysis than others. And indeed, there were a couple of very good enzymes that gave the yeast a good capability to degrade the oligosaccharides and released more carbon sources for the formation of lactic acid.
Which yeast strains were engineered in the project?
We were working with a couple of different yeast species. So first we worked with yeast Saccharomyces cerevisiae, which is the baker's yeast, and then we also worked with the Pichia kudryavtsevi yeast, which is not as commonly used, but it is an industrial production organism also, and it's particularly acid tolerant.
And what about the timeline of the project?
Project as such has been going on for roughly 4 years, and already early on we generated the yeast strains, or the first yeast strain, and I think that the sort of main breakthrough was when we first saw something that was made of the lactic acid that was polymerized to polylactic acid, and there was this small spoon made of the polylactic acid from our yeast. So it was somehow a very concrete demonstration that yes, it can be done. But yeah, the yeast construction, the genetic engineering part is not very time consuming. Lots of strains can be generated rather quickly. It's like the timeline is weeks or months rather than years. But then after we have the engineered yeast, there is a very lengthy and extensive phase when the cultivation conditions are optimized So first we start the cultivation in small scale in shake flasks, and then the cultivations are gradually scaled up into bioreactors. So the cultivation volumes are like, let's say first 1 liter, 10 liters, 30 liters, 100 liters, and eventually the largest scale cultivation was 1000 liters, so one cubic meter. And the good lactic acid production properties were retained. Also in the large scale cultivations, there has been more than 10 large scale cultivations done in the 1000 liters, and from those cultivations the lactic acid has been purified really in a large scale. So from this 1 cubic meter fermentation we get roughly 100 kilos of lactic acid, which is then purified in order to remove unwanted compounds that may interfere with the polymerization of lactic acid into the polylactic acid. And there are lots of impurities in the soy molasses, which is like a dark syrup and still contains carbohydrates, acids, ions, which have to be removed in order to make a high quality lactic acid.
Once the lactic acid has been produced from the soy molasses, it is polymerized at Bright Plus to create PLA. This PLA is made into a foam which can replace the thin foam's XPS polystyrene insulation.
XPS on tätä sulkepullistettua polystyreeniä, ja tämä PLAhan on hyvin niin kuin polystyreenin kaltainen muovi, ja sen takia se niin kuin oli meillekin sit se paras vaihtoehto. Tärkeämpää, että se muovi on kierrätettävissä helposti kuin se, että se on niin kuin nopeasti biohajoava.
Our next waste material is also surprisingly abundant. This sugar source is pectin. Annually, hundreds of thousands of tons of fruit peel seeds and sugar beet pulp, a side stream of sugar production, pile up without high-value utilization. Currently, this treasure is mainly a problem. It is used for landfills or dried for cattle feed, which is quite costly. In this example, we would like to demonstrate a biotechnology case in a chain of technologies patented by VTT, where we can turn this pectin feedstock into platform chemicals, which in turn can be further processed for numerous applications. To learn more about this exciting research, check out our recording. We presented also this case in our previous webinar, and you will hear the full story how a consolidated process was developed. Our next waste material is also surprisingly abundant. This sugar source is pectin. Annually, hundreds of thousands of tons of fruit peel seeds and sugar beet pulp, a side stream of sugar production, pile up without high-value utilization. Currently, this treasure is mainly a problem. It is used for landfills or dried for cattle feed, which is quite costly. In this example, we would like to demonstrate a biotechnology case in a chain of technologies patented by VTT, where we can turn this pectin feedstock into platform chemicals, which in turn can be further processed for numerous applications. To learn more about this exciting research, check out our recording. We presented also this case in our previous webinar, and you will hear the full story how a consolidated process was developed. One of the most abundant industrial side streams in the world is of course CO2. Using this carbon source efficiently instead of pumping it into our atmosphere is no doubt one of the most important areas where synthetic biology can help. The quest for microbes naturally capable of using this abundant raw material takes us to the very beginning of our planet. At Aalto University, Professor Silvan Scheller's team is working with an intriguing group of microbes, methane-producing archaea.
Question. So they actually come from the time before there was oxygen on our planet, and they probably originate even before photosynthesis. And so this was actually a time where there was much more CO2 in the atmosphere than what we have now. And these organisms, they have learned to actually eat the CO2 and then with the help of hydrogen convert that CO2 to methane. And basically by producing a fuel, they can extract energy from that process. And so that's quite remarkable. And so nowadays you find those at sewage plants or in the rice fields or in the rumens of cows, or a few inside you maybe also. So they are at places where there's no oxygen because they're actually oxygen sensitive. And that's actually why we use this laboratory equipment where we work with the exclusion of oxygen, so to keep them in their environment that they are able to live, basically.
These organisms could serve as a microbial alternative to the Fischer-Tropsch synthesis in a power-to-X plant.
In the future, the microorganism can also convert electricity and CO2 to ethane, because the electricity is not easy to store. But if we convert it into ethane, then that means we are storing electricity in a gas.
Scheller's team is reprogramming these ancient organisms to produce ethane instead of methane, so it can be used in diesel engines.
I mean, the main change is to convert the CC bond. So because the natural methanogens, they convert CO2 to methane. So this is from a 1 carbon to a 1 carbon compound, but it corresponds to an 8 electron reduction. But now if you want to produce ethane, ethane has 2 carbons, so you have to form a CC bond. And the methanogens, they can already produce acetyl-CoA, so this is a C2 unit, but they use that only to grow and not to produce ethane. So what we want to do is to direct the carbon flux fully to this acetyl-CoA, and then actually use this acetyl-CoA to produce ethane from that. And this needs implementation of new enzymes to achieve that, basically. There is different ways to do that. So one way could be enzyme engineering, so to change them from C1 to C2 substrates. But in fact, there are closely related microbes known that can eat ethane and produce CO2. So basically microbes that do the opposite process, they exist. They have been discovered in 2019. And basically we use the same pathway but in the opposite direction. So that means we can actually take the genes from these environmental microbes and implement them in our methanogens to achieve these C2 transformations. So this is actually, I would say, an interesting application of synthetic biology, that you can go to environmental microbes, take their DNA, and then use it to build up a new system, or in our case, the reverse direction of the metabolic pathway. We can have renewable energy converted to electricity, electricity to hydrogen, and then hydrogen and CO2 to C2. But the final goal would be to have C10 or so, like what we use for jet fuels.
But why use such an exotic microbe?
And so instead of just constructing the whole thing always in the same microbe, my approach is to use the microbe that is already very close to the process that we want to make. And then we need not so many steps to do that. Like theoretically, you could try to make the whole ethane project in E. coli, but this will most likely never work. I mean, there's first of all hundreds of genes that you will need to implement, and then you have another redox potential, so the whole cell is not meant for producing methane. But then this archaea, they evolve to produce methane. So for them it should be a smaller step to go from methane to ethane.
Around 2 billion years ago, something extraordinary emerged from the primordial seas of our planet. Photosynthetic bacteria. Known to many as the annual blooming blue algae, the cyanobacteria generated the Big Bang of life, turning the atmospheric CO2 into oxygen and thus enabling the explosion of life above sea level. At the University of Turku, Professor Jagut Alaverdieva-Rinne and her colleagues are reprogramming these bacteria to produce valuable chemicals.
Photosynthesis is the most unique process in nature. The photosynthetic organisms are able to capture light photons and use that light photons to split water to electrons and oxygen. That electrons are used to stimulate photosynthetic electron transport to produce NADPH and ATP, which are later on powering CO2 fixation. So for this process, you don't need anything else.
The aim is to use this wonderful natural process to produce chemicals that are currently derived from fossil-based raw material or land-grown biomass.
Photosynthetic algae and cyanobacteria as a production system has advantage over plant production systems, and this is related to the fact that they have a very short doubling time. Some cyanobacteria can double in 2 hours, whereas for plants you need days, months, years.
Koska levät on usein yksisoluisia, käytetään, niiden ei tarvitse tehdä juuria ja runkoja ja tällaisia, vaan että se varastoituu siihen samaan soluun se auringon energia kemiallisessa muodossa.
In the case of photosynthetic organisms, the structure of the photosynthetic machinery is of course known, but I think it's about controlling it. It has been developed in evolution over millions and millions of years, and then we come and take the machinery and try to modify it to our use. And it's not simple, it's very complicated. The system is so effectively buffered against different types of changes, and we need to understand those buffering mechanisms, and we need to understand how to overcome the biological mechanisms that are trying to protect the system. So in a way, we are battling against the biology of the cell so that we could take it to our advantage, while the cell is trying to just protect itself, more or less.
Our aim in this project that to use photosynthetic cells not as a biomass for biorefineries, but as biocatalyst, which can convert solar energy and CO2 into the targeted molecules. And here we need also synthetic biology tools, because cells don't produce all the compounds we are interested. We can use emerging synthetic biology technology to bring foreign production pathways to the cells and force them to produce the compounds, chemicals we are interested.
Photosynthetic organisms are primary producers. They use solar energy and CO2 to produce organic compounds, whereas heterotrophic organisms such as E. coli and yeast require external organic compounds like sugars to operate. This gives photosynthetic organisms a clear advantage.
Many of the heterotrophic production systems, directly or indirectly linked specifically to the TCA cycle, the energy conversion system which actually takes the energy of the reduced organic compounds that the organism is using. However, in the case of cyanobacteria, we are not so much interested primarily in the functionality of the TCA cycle, but instead how the metabolic flux is diverted from the photosynthetic machinery towards the downstream reactions. In the case of ethylene, this is different, because ethylene production pathway is utilizing specifically 2-oxoglutarate, which is an intermediate in the TCA cycle, as a precursor. So in this case, our question is a little bit different, because our question is, how do we force as much of the metabolic flux as possible towards the TCA cycle, while in the normal scenario, in the nature, this flux is very limited.
The researchers at the University of Turku have extensively been exploring and developing ethylene production in cyanobacteria.
And the basic concept is that in nature there is a specific soil bacterium, Pseudomonas, which is capable of producing ethylene. And that specific bacterium is producing ethylene because it's a plant pathogen, and it uses ethylene to trick the plants, because ethylene is using as a signaling molecule in the plants. So the bacterium takes advantage of, tricking the plant more or less. So what we have done in this field is taking a specific gene which is associated with producing ethylene in that specific soil bacterium and putting that one single gene into cyanobacterium. And just this one gene enables this TCA cycle intermediate 2-oxoglutarate to be converted into ethylene, which then can be collected. That basic concept is very simple, but the entire reaction network that is associated with this is very complicated. We have just released a review article on this, which tries to combine the information that is currently known. And in the end, although the single ethylene producing reaction is very simple, there's really a lot of things going on inside the cell. And we have looked at this ethylene production from different angles. And one of our studies was looking at this ethylene forming enzyme, that specific gene and its regulation, so that we were trying to find optimal regulatory systems for that specific gene, so that this genetic information could be first transcribed into RNA and then translated into protein, which is the actual effective motor that catalyzes the reaction. So our viewpoint to this, our approach to improve the ethylene production system, is based on the fact that we have optimized some of the regulatory mechanisms for this F gene.
However, there are still many bottlenecks in scaling up photosynthetic ethylene production on an industrial scale with traditional photobioreactors, such as high production costs and low productivity.
One of the major limitations of suspension cultures is their low light utilization efficiency. In the dense algal cultures, we have a significant problem with low light utilization efficiency, with the self-shading effect. The internal part of the bioreactor doesn't work very efficiently, and the overall efficiency of the production is very low. We have a solution to this problem. So we distribute the cells within thin layer immobilization matrices. This significantly improves light utilization efficiency and also decreases water consumption. And now we combine this technology with engineered cells and call this technology solid-state photosynthetic cell factories.
An additional aspect of the thin layer architecture is to engineer different types of strains with altered light penetration properties. The photosynthetic cells have light harvesting antennas to capture the photons. However, these antennas containing green pigment have a self-shading effect in photobioreactors. Perhaps counterintuitively, by shortening the photon capturing antennas, more efficiency can be achieved in overall light conversion.
We will have the two layers of the cells with the normal antenna and with truncated antenna at the top. And in that case, the light will penetrate easily through the culture with truncated antenna and becomes available to the culture with the normal photosynthetic antenna.
The concept has been successfully demonstrated in a recent publication.
Although there are several proof of concept studies for the production of green ethylene, these modified cyanobacteria could produce ethylene only for a few days. But in our recently published Green Chemistry journal, we employed a thin layer artificial biofilm with embedded cyanobacterial cells that can produce ethylene for a longer period.
The results have been very promising for both cell viability and production yield. Ethylene was synthesized from light and thin air.
These artificial biofilms can act as a true photobiocatalyst, and they can produce ethylene for up to 40 days with high efficiency of light to product conversion, and the yield is 2 times higher than the suspension cultures.
To improve the immobilization matrix, the researchers in Turku have teamed up with material scientists at VTT to create a superior nanocellulose-based culture medium.
We hope that we can, I would say, revolutionize the solid-state cell factories in a way that we are getting out from these conventional dispersion cultures to solid hydrogel layer or film type of cultures, where we can increase the cell density and decrease the water intensity of the system, and also to make these cells live longer, improve the cell fitness, and improve their productivity.
This research is part of a FED-funded research consortium, Futural Leaf. The aim is to create a matrix with an architecture that mimics the natural leaf.
About the green leaf, it has layers. It has a very, very dense layer at the top, which allows light penetration, allows gas exchange, keeps out moisture or keeps in moisture, and then when you go down, you have something that's really porous, and again, exchange of gases, nutrients within the material.
The team at Turku is also developing a bead-shaped alginate culture. This shape is helpful when producing sucrose, which requires continuous shaking and high salt concentration in the media. The sucrose is produced as the cell protects itself from the osmotic pressure caused by the salt.
These cells are synthesizing sucrose in a natural pathway when in an environment with high salt concentration, and with the genetic modification we are capable of making the cells to secrete the sucrose into the growth media. This sucrose can be used for feeding other heterotrophic bacteria like E. coli or yeast, which can be cultured together in the same bioreactor, in the same media with the beads. They can also be co-immobilized in the same matrix, or this can be separate reactions as well.
Vilja Siitonen is producing yet another end product using genetically modified green algae.
The targeted compound that these cells are producing is epsilon caprolactone, and it is used as a polymer precursor at the moment, and it typically is made in an unsustainable way, but this way how we are making it with the photosynthetic cells, it would be sustainable. In my case, I use Chlamydomonas reinhardti, and the benefit of using eukaryotic algae is that the things are compartmentalized. So we have different cell organelles where different things are happening. So we can modify the photosynthesis, and at the same time the respiration is not affected necessarily. And this gives us valuable opportunities to work on the modifications and make the photosynthetic activities better and more effective.
What kind of modifications have been done to the cells?
I have introduced this cyclohexanone monooxygenase, which makes the cyclohexanone to epsilon caprolactone, and the gene is from Acinetobacter species.
Moving from fundamental research to commercial scale, Nordaqua is paving the way for industrial applications of micro and macroalgae in the Nordic environment. This multidisciplinary Nordic Centre of Excellence is funded by Nordforsk Bioeconomy program and coordinated by the University of Turku.
We aim to bring algae and cyanobacteria photosynthetic platforms a step further into industrial applications. For instance, to reduce the algal production costs, we use an hydroponic greenhouse effluent to generate biomass.
The nutrient-overloaded greenhouse effluents are a major culprit in global eutrophication of natural water systems. In a recent publication, solutions to this problem was explored harnessing microalgae.
Hydroponic greenhouse effluent generally circulated several times and after then discharged into the environment. By using this effluent for growing algae, we create a closed loop. We generate biomass, valuable biomass, and also we recover nitrogen and phosphorus.
Next, we will continue looking at utilizing CO2 as a feedstock. Another group at VTT is studying an entirely different group of carbon-fixating microbes, the Canal Gas Bacteria.
They are actually very interesting species. There are lots of different bacteria that can utilize carbon dioxide as their carbon source, but Canal Gas bacteria are a bit different from those. They actually utilize for carbon fixation Calvin cycle like plants and algae, but instead of using sunlight or getting their energy from sunlight as plants and algae do, they use hydrogen to run their metabolism. So they are bacterial species that fix carbon dioxide via Calvin cycle and use hydrogen as their energy source. So at VTT we have studied a couple of these Canal Gas species already for a while, and in general they have been studied a lot for production of nutritious single cell protein for food and feed purposes, but here at VTT we have also been interested to explore the possibility to use them for chemical production from carbon dioxide. In our recent publication we actually showed the production of lactic acid and beta-alanine with Rhodococcus opacus, that one of these Canal Gas strains. And lactic acid and beta-alanine are the products that both can be used to make biopolymers. So lactic acid is already used for making polylactic acid, and also beta-alanine can be deaminated, and as a result the precursor of, for instance, nylon 6 is produced. And in this article we show that these compounds can be produced from carbon dioxide directly with this Rhodococcus opacus, and we also demonstrate that they can be produced in this kind of bioelectrofermentation where the hydrogen needed as energy is produced in situ in these fermenters via electricity, and then carbon dioxide is fed to the fermenter from outside. We have now demonstrated the production of these compounds at the proof of concept level, but I believe that the rate and the yield can be increased with further engineering of these bacteria, and of course there are already examples of how this kind of bioprocesses, these gas fermentations with these Canal Gas bacteria can be scaled up, so it's possible.
The Canal Gas bacteria are also the stars behind Solar Foods protein production. VTT has developed for many years applications in cellular agriculture, generating several spin-offs.
I think the two spin-offs from VTT, the first one is Solar Foods, spun out 2017. So they are making Solane, so that's really the single cell protein for food, so using carbon dioxide as a feedstock for the microbes. So it's very nice case also for the sustainability, so it's a nice case. Then we have another single cell protein production spin of Enifer Bio, and their idea is to produce feed, fish feed actually, and they use industrial side streams as a feedstock also in their production, but they are liquid side streams and different strains, of course. But it's also a nice case because, of course, the transition in the food production, it will not be sharp that we stop eating meat and like that. So we also need sustainable feed sources. So Enifer Bio is targeting at that and soy replacement. So it's a nice case. Then where we are working towards commercialization is the egg white production by Trichoderma reesei fungus. So we have won also the Aalto Innovation Prize there. So we have got very nice results in producing this egg white protein by this VTT fungus Trichoderma reesei. And then one theme we are working at VTT for food production is plant cell cultures. And that the idea there is that we can produce certain, let's say, plants that are maybe not grown so sustainability at the moment, avocado or cocoa or plants like this. So we could produce them in cell factories as well and get the enjoyment of those plants by these cell culture technologies.
One of the challenges with microbial food production is the misconception around GMO food. Microbially produced food is not genetically modified, rather the microbes that is producing it. And those microbes are of course not grown in nature, but in closed industrial fermenters.
Both the consumer communication and consumer acceptance and then the regulatory issues, they are both super important things for the cellular agriculture, and we need to make good things there. And if we first think about the consumer communication, of course I think we have to be very transparent how we do it, but we can of course highlight the good things of cellular agriculture, like ethical way to produce and safe way to produce food, because we don't need the animals, for instance, anymore. So it's really thinking about that aspect, so it can be a good asset. And I think we have been already seeing the signs that, for instance, I just saw a report where the cultured meat was asked by the consumers how do they feel about it, and it was not like no-go. There is in a way the positive feeling. So I think the next generations are really more open to the new food innovations as well. So, but still, I think consumer communication, it's really important, and we have to be doing it wisely and transparently and explain what we are doing. Then about the regulatory issues, that's of course something to be considered, and we really need to take care of that. Especially in Europe, there is of course these products most often they fall into the novel food category. So there is the steps to take, and that's something we just need to do to ensure that the food is safe for the consumers. Then of course when we go to the GMO, genetic modification of organisms, and that regulation, so there I hope that European Commission will be in a way open-minded and re-evaluate the decision they have made in the technologies for how to modify organisms and what is safe and what is not. But of course, when you talk about food, the safety is the key thing. So we always need to evaluate the safety. That's super important.
Harnessing microbes for industrial purposes using synthetic biology requires a systematic design, build, test, learn cycle approach, utilizing all the available tools on top of traditional lab work, from computational modeling to automation, robotics, analytics, and machine learning.
To achieve this, first we design the cell using computational tools to have a model of how to build the cell and in our case usually a microbe, as we think it would work optimally. Then we build, that is, we engineer the cell according to the model using genome editing tools. Because with current knowledge our designs are not perfect, we need to test a large number of engineered strains, how they behave and what their cellular chemistry is, and finally we learn from the data to design improvements.
At VTT, the infrastructure and know-how in biotechnology and synthetic biology build on decades of experience and excellent in-house facilities from lab to pilot-scale testing. Before doing wet lab trials, synthetic biology offers tools for predicting the outcome of metabolic engineering of an organism. This includes a fascinating area of computational tools, analytics and machine learning for engineering biology. At VTT, Paula Johten's team is specialized in this highly complex field of science.
Our team at VTT, we combine several kinds of expertise. We have computational expertise, so we do this data-driven modeling, and we do mechanistic modeling. So these tools help us to handle the complexity. So biology is dynamic, complex, these interactions that take place for some moments and then disappear. Biology is so highly complex that we could never ever cover everything just by exploring with high throughput experiments. For all this complexity we need help from both these mechanistic models as the metabolic models, but also these data-driven, so the machine learning models. And we believe that the combination of these models will be the success story in this field to develop learning cycles, and so that these computational approaches will be in the future efficient in suggesting the next modifications to cells and take into account the different kinds of functionalities in cells.
The modeling can significantly limit the number of wet lab trials as they are able to predict cell metabolism.
So these genome scale metabolic model simulations, they kind of give or take into account the ultimate limits of what is possible with the particular metabolism that the species has. So it is based on the ultimate laws of mass conservation and thermodynamics. So both of these become taken into account in the simulations. So we can handle with computational means in a predictive sense. So we can do predictions on what kind of chemistries the cells should be able to perform and how efficiently. So what should they contain? How efficiently should these reactions take place when they are metabolically active to meet our aims with the applications?
This method is especially valuable when venturing beyond the well-known model host organisms, such as the yeast Saccharomyces or bacterium E. coli.
For model organisms, there's a lot of accumulated knowledge, functional annotations, phenotypes recorded and such. But as we want to exploit the wide variety of natural species, we want also to be able to do this predictive modeling for other species, the non-model species. Many of them can be very interesting for applications. They have capabilities that the model organisms do not have. So what we can do is that all the information is there in their genomes. Genome sequencing is fast and affordable nowadays. So we generate these genome sequences. We functionally annotate the genome so that we can gather information of which enzymes are these species able to encode. So what biochemical reactions can take place in those cells. And then we assemble those into the network. So we do metabolic model reconstruction for any species from sequenced genomes.
But how do you derive new functionalities from the extensive sequencing data?
So there are huge sequence resources nowadays available, but we need to be able to there choose then the correct sequences that would have the functionality that we want to introduce into the cells of interest. And so for this reason, we need to understand how does it translate into functionalities. And so here we have done like conventional bioinformatics for tens of years already, and so this is efficient. So there's a lot of work put into also characterizing genes, and we use all this accumulated information on characterized genes and gene products, and we use this to identify new similar that could have similar functions but maybe different characteristics, maybe more efficient enzymes of the same kind, those ones that can convert the same substrates into products but more efficiently. So these are the enzymes that we look for in the sequence resources. But as we know, so there was a major breakthrough in the end of last year, so this AlphaFold 2. So you know, the protein functionalities are highly dependent on their three-dimensional structure. And this whole folding problem has been a major problem or challenge in life science research. And so now there's a computational approach to predict the protein structures from the sequence. And this makes it possible then to use these structures that are the basis for the functionality. So we can use even these to identify novel sequences that could have the functionalities that we need for the applications.
Once the new cell functions are designed, the new genetic information, DNA, that is required for the desired functions needs to be constructed in a form that it can be inserted into the host organisms. So-called expression cassettes contain the genes and their regulatory functions.
The expression cassette is typically a promoter, then a gene and a terminator, which if you put it into a host, the promoter will harness the transcription, activation of the expression of this gene.
Once the vector is designed, the individual parts can be ordered online.
It has a huge options. It's a fully synthetic DNA, so you can actually order nowadays. There are companies which are actually making this fairly cheap, so you can order whatever DNA sequence you want, and they will deliver in a small tube DNA of your choice.
Once parts are assembled, there are several gene modification tools, such as CRISPR, to edit or insert the foreign genes into the host organism. At VTT, a fully synthetic expression system called SES has been developed for expressing genes in a huge variety of different eukaryotic hosts.
That expression system we developed a couple of years ago with the aim to actually be able to express genes in diverse organisms without need to use native promoters, native DNA parts of that particular organism.
The core of the system is that it is based on a universally active promoter element which enables the expression of all the subsequent parts of the system. So when the core promoter controls expression of the synthetic transcription factor, then any cell can express the transcription factor. This transcription factor then binds to DNA elements in a chromosome where we have engineered the binding sites for this transcription factor. The binding of this transcription factor to these binding sites then triggers the activation of the target gene that is under the control of the SES promoter.
This together forms the binding sites, and the core promoter forms a new synthetic promoter, new to nature. It doesn't exist in the nature, but it's functional in the fungi or in eukaryotic organisms.
And the important thing with the SES system is that we can not only express at very high level, but we can also tune the expression level. So from very weak to gradually to very high, higher than the strongest endogenous promoter. In addition, our experiments have shown that the SES system is very stable and it maintains the expression levels also in bioreactor conditions. So the set and designed expression levels are maintained throughout the bioprocess. An exciting feature of the CESS system is that due to its modular structure, we've been able to use different features of the CESS system to build complex genetic control circuits in cells. These circuits make use of DNA elements that enable either activation or deactivation of gene expression. Due to modularity, the CESS system is well suited for high-throughput approaches using robotics. By using CRISPR technology together with CESS modules, we can generate a large number of strain variants where the selected target genes are expressed at different levels. By combining these properties in different ways and targeting them to control key nodes in the metabolic network, we can direct cell metabolism for a designed end result. We have shown that it is possible to build genetic circuits that have a memory so that they enable cell metabolism to stay in a given mode even after removal of the short triggering signal.
Once the desired strains have been constructed, the automated testing facilities at VTT can be used, ranging from microtiter to well-controlled bioreactors of different sizes.
For the test phase, we have various automation platforms where we can test the strains in various formats. So we have the screening robot system where we can test the strains in a microtiter plate format. Then we have a biolector, which is a bit larger volume, and there also that's in the a bit larger microtiter plate. And then we have an Amber 250 system, which is then a bioprocess automation system, where we have 24 bioreactors, and there we can also test the strains in a larger scale, which then resembles traditional bioreactors, which then also represent the pilot scale and the industrial scale that we aim to do.
We can carry out benchtop bioreactor experiments, so those are typically ranging from 1 liter up to 5 liters. We have standalone reactors, 10, 20, 50 liter scale, and then also the piloting equipment, and that's ranging from 50 liters to 1,000. And then we have quite well equipped downstream processing equipment system downstairs, so we can do cell separations, filtrations, up concentrating of broths, and various types of downstream processing steps there.
When scaling up, what kind of challenges are met when dealing with larger volumes?
Especially when we talk about process scale-up, there are several things we need to take into account when the reactor size is increasing, like mixing is a typical issue in the process scale-up, and aeration. Microbes, often they require a lot of oxygen input, and that can be challenging when you scale up the process. And then we have different types of side streams we are working with, so pre-treatment steps before you can actually ferment your microbes. Then we think about other like purification related costs, so that's often like something that you don't really need to think about that much when you do experiments in small scale. But then in our pilot facility, then these are the things that we really need to see that the workflow works well. And for that, at VTT, we have quite unique platform to carry out all those different steps.
The analytics team at VTT have decades of solid experience and can carry out a multitude of measurements.
We have the bioanalytics and small molecular analytics in the team. So we can do fast measurements, we can do extensive measurements, we can do targeted analytics that are like, suited for certain applications. We develop novel methods there.
After testing, the DBTL cycle continues to analyze the experiments and incorporate what has been learned into the next cycle. And the basis for the learn phase is all the gathered data.
The high throughput power, that's a very cool thing that we can have so many individual experiments simultaneously, and we can gather so much data out of one day of work. And then if we go to the larger scales, and then we can measure various things from the same experiment. So for example, in the Amber system, so we can measure all the things that you can measure from a traditional bioreactor, and we can also take the samples automatically. So we can have a sample every hour for let's say 7 days. So we can also there gather a large pool of data that we can then utilize in the DBTL cycle.
We are really looking forward to see like fast DBTL cycle, because this allows us to accumulate the data so that finally we can employ so efficient machine learning algorithms to guide this engineering process. So some of these approaches are already possible. So we can apply machine learning to help the scientists by suggesting new modifications to the next round.
So Paula, what do you think are the expected benefits of the interplay of human scientists and machine learning tools?
Yeah, I think we'll become faster. We'll become faster because by the scientists looking into the data and thinking, so we are clever, but we are slower. So if we can combine, like maybe even this combined kind of human-guided machine learning, that sometimes the scientist can interfere and take part in the learning cycle, but that the machine learning algorithms will enable us to do this faster, and this will be of course then supported by the automated means for engineering cells.
And if you think about the big picture, what does it truly entail?
I think we should understand evolution better. So the possibilities of the evolution, and it has several kinds of effects on biotechnological applications. So on one hand, we would like to have our engineered functionalities to become stable, that they would function there, that they would not get changed by selection and by other evolutionary forces. And then on the other hand, evolution has enabled formation of all these super exciting characteristics that we see in nature. So could we actually use this better? So if we say that we could take into use all the functionalities that we have seen in nature, but what if we could actually use these forces to develop something new? Something that we can use for the applications. So I think this will be a big theme in future, like synthetic evolution.
Scientists at VTT have demonstrated continuous production of mycelium leather at 1 meter per minute. The material is 100% bio-based, and production is environmentally sustainable. Fungal mycelium is produced in a bioreactor, collected and processed. The mycelium can then be casted continuously to produce a film. Finally, the sheet is dried, resulting in a leather-like material. This new material can be colored and patterned, while the production method ensures consistent material quality with competitive production costs. Developing alternative sustainable materials for the fashion industry is a major environmental concern. The industry is responsible for a full 10% of global carbon emissions. That's more than all international flights and maritime shipping combined. And a major culprit within the fashion industry is leather production, with a carbon footprint estimated to be equivalent to the CO2 emissions of 30 million passenger vehicles annually. Although leather-like materials can be made from bio-based and animal-free resources, no one has created a leather substitute that is sustainable, cost-effective, and meets consumer expectations. Researchers at VTT believe biotechnology could be the answer.
So we grow fungal mycelium and use it to make leather-like fabrics to replace animal leather.
When we look at the fashion industry and the leather industry, it belongs to a really complex supply chain. So I think by looking at these new forms of alternative materials to leather, like those produced with fungi, I think it can really change the industry.
All filamentous fungi have mycelium. Mycelium are the root structures of filamentous mushrooms. And this is what scientists engineer to produce materials.
We control the conditions so that we only get the mycelium. We don't need the mushroom.
Synthetic biology allows scientists to design materials from the cellular level. This enables them to achieve a variety of desired properties.
We can start to bioengineer mycelium and play with the strength, tear resistance, abrasion resistance, try to bring in new functionality into the material by bioengineering methods.
In addition to its superior performance, the new material is significantly more sustainable than leather in many ways.
We can control and make the mycelium grow from, let's say, organic waste, for example, and because we don't need fields, we can grow it in the lab conditions. We save surface land area. We don't produce so much carbon dioxide as with the cows, for example, and end product is biodegradable.
The new leather project brings together designers and scientists to tackle the challenges of conventional leather production.
And I think as designers, we understand like the end consumer and their needs. So then we can then bridge this gap to create products that actually are needed by the consumers and at the same time are responsible to the environment.
The demand for sustainable materials is growing rapidly, and new leather takes us one step closer to a more sustainable fashion industry. The textile industry weighs heavily on the environment. For instance, it accounts for 1/5 of all global water pollution, and 99% of the pigments currently used in textiles are fossil-based synthetic dyes. To address this problem, the Biocolor Consortium, funded by the Academy of Finland, is looking for answers from nature. Satu Hilditch, a researcher in the Biocolor project, takes this challenge seriously. Together with a team at VTT, she is searching for a solution that utilizes nature's own synthesizing power to create pigments. Natural dyes produced by plants, insects, fungi, such as mushrooms, and even snails have been used for thousands of years. But collecting the dyes from nature is not very practical or scalable. Nor do we have enough arable land to cultivate pigment-producing plants to meet the entire global demand. However, biotechnology can overcome these bottlenecks. Using microbes instead of plants or forest mushrooms, pigments can be produced in bioreactors without using arable land all year round. Another important benefit is that microbes produce pigments in a benign water-based system in mild conditions. Therefore, harsh chemicals can be avoided. Utilizing the tools of synthetic biology, the ability of natural organisms to produce dyes can be transferred to microbes which are suited for bioreactor cultivation.
Now we have a possibility to make a genetic modification and take only the genes that are responsible for the colors and express them in a host organism that can easily be grown in larger scale, so we can produce a lot of color in this production host.
Like all production, the production of dyes needs a carbon source to start with. With microbes, this carbon source can be obtained from sugars or even industrial side and waste streams.
For example, from food industry as carbon source for baker's yeast to make the bio-based colors. And in the future it could even be carbon dioxide that we could use as a raw material for colors.
Many fungi produce different colors. There is the whole spectrum of different colors can be produced by microbes. And it's just a matter of making the process more efficient.
The diversity of natural colors and their properties are immense. Furthermore, future possibilities go beyond visual properties.
Some of them being possibly antiviral or antimicrobial and or UV protective, and they have different properties in their stability, or possibly in the future we could even enhance some of these properties. So create kind of designer molecules with desired substitutions, for example.
All biochemical reactions in nature are catalyzed by enzymes. The genes are the code for making these enzymes, and they are also responsible for the production of pigments. The aim of Satu Hildich's work is to find and isolate the specific DNA sequences, the genes, which dictate the production of specific colors in natural organisms.
Colors are secondary metabolites, and those genes that are responsible for those colors are often packed in clusters in the genome. And so I have a hunch which type of a gene I start looking in the genome, and then when I spot it, then I can find the other genes around it that are also involved with the color pathway.
Although some organisms have their genome sequenced, the world of genomes is a vast, still partly unknown territory for researchers to explore, and synthetic biology is offering new tools that enable going farther than ever before.
It's really exciting to find new genes that you have a feeling that nobody else knows this. There are so many genes to be found, but then still when you find it and clone it and see that this gene is really making this enzyme and this enzyme works, then it's quite cool that you go home and think, I know something that nobody else knows.
Doctoral student Robert Pylkkänen at Aalto University, the Center for Young Synbio Scientists, is researching how ordinary yeast can be used to produce cellulose without trees or other plant-based biomass.
We're making polysaccharides such as cellulose or beta-glucan, and these reactions can be carried in a test tube, so we can make cellulose in a test tube, which is quite exciting.
In his research, Pylkkänen has used an enzyme that can build cellulose polysaccharides from sugar monomers. In nature, this enzyme does the opposite job, degrades wood. This enzyme was found in an extremophilic bacterium discovered in volcanic soil on the island of Izu in Japan. The research was originally launched as a Finnish-Japanese cooperation project.
Enzyme was isolated from extremophilic bacteria, and what it did differently compared to other bacteria is that instead of using water to break down cellulose, it turned out to be a phosphorylytic enzyme. So it utilized phosphorylases to break down cellulose, and then the difference there is that instead of breaking down the polymer to glucose units, we actually break it down to glucose 1-phosphate. So we retain in a way the energy of the glycosidic bond. And then also this enzyme can be used for the reverse reaction. So if we incubate it in the presence of glucose 1-phosphate, we can make cellulose.
Pylkkänen does not produce the enzyme with the exotic bacterium, but he has transferred the genes that control the production of this enzyme from the bacterium into baker's yeast. The material produced by the enzyme amazes researchers. The sugar chains and the hexagonal three-dimensional structures it forms are completely unique and nothing like it is found in nature.
First you have the individual glucose chains that are making like this hexagonal chain, so it's always 6 glucose units per one turn around the helix. Those polymer chains, they come together and make larger structures, and also those structures are hexagonal in shape. But then we see that even those structures come together to make even bigger hexagonal structures, so they are making like multiple layers, and you have huge 10 micrometer in diameter hexagonal structures in the end, but it all starts in the nanoscale.
While it is not yet known what all these new materials could be used for, the possibilities are exciting. For example, they may be suitable for medical use or used as a paint additive, replacing fossil raw materials.
When we synthesize it, like starting from nothing, then it's quite interesting to think that you could make the cellulose in a very tailor-made fashion for any application. Of course, it's like a long way ahead. But already, when we have total control to the synthesis, we can, for example, modify the reducing end and then use that like starting molecule to make then cellulose polymers that have the same modification. So, for example, if we modify the reducing end and then of glucose and use that molecule to make cellulose chains, then all those cellulose chains would have the same modification. And it's not so easy to modify cellulose that's already 1000 units long, but when it's like in smaller pieces, then it's much easier.
Japanese researchers recently used this enzyme to produce nanocellulose in space, in an experiment at the International Space Station. The method worked well there because it requires no heat or hazardous chemicals and takes little space. The production of cellulose in space, in restricted conditions, can help us understand what kind of solutions sustainable industrial production really needs in a circular bioeconomy. Doctoral candidate Anna Ylinen is developing biopolymer production in yeast.
And I'm using synthetic biology methods to adjust the properties and making the production hopefully cheaper in the future. I'm producing especially PLA, which is polylactic acid, and then different copolymers with that, also polyhydroxybutyrate, PHP. Usually the lactic acid itself is produced with microorganisms and then it's purified and polymerized with chemical methods. But now we are producing both the lactic acid and the polymer already in the same cells in vivo.
Anna's research is based on the activity of various engineered wild-type PHA synthase enzymes. PHAs, or polyhydroxyl alkanoates, are natural polymers made by bacteria found both in soil and marine environments.
They are completely watertight, so it looks and feels like a normal plastic, but it's biodegradable and compostable.
Ylinen has transferred different bacterial and fungal genes to yeast in order to broaden the feedstock base.
I'm modifying the yeast cells that they would learn to use cellobiose as a raw material instead of glucose. So we would want to use sugars derived from, for example, from cellulosic biomass instead. I've produced polyhydroxybutyrate, BHB, with cellobiose, and I've compared two different enzymes inside the yeast cells. That one of them is using the cellobiose a bit more efficiently and faster, but the other one would have more beneficial features regarding how the cell divides the energy inside it. So we are kind of trying to find the optimal way to use the cellobiose for these purposes, but also for other applications.
Ultimately, this could even entail using industrial side streams as raw material.
The bacteria cells are efficient of what they do already at the moment, but yeast cells have a lot of potential for using industrial waste as a raw material, so for using new different carbon sources, since yeast tolerate low pH and harmful compounds very well, so maybe we can take directly some industrial side stream and we don't need to pre-treat it that much in the future.
Finally, we will meet Peshmerga Mohammadi, whose recent publication has attracted a lot of excitement. Peshmerga is a researcher here at VTT, and his work has been funded by the Vihuri Foundation's Center for Young Symbio Scientists. He is exploring entirely new classes of advanced biomaterials. One source of inspiration has been the mantis shrimp, which is an amazing tropical creature that lives in shallow waters. Not only does it have absolutely exceptional eyes with 16 types of photoreceptors, but it also perceives polarized and ultraviolet light. On top of that, it has the world's strongest naturally occurring material as its weapon.
It's a material that has a really high mechanical strength,
and high impact resistant. So in this work, our aim was to look into a possibility of mimicking some of the most essential features of the club to create a new type of nanocomposite material never described before.
The blow of a mantis shrimp corresponds to the power of a small caliber weapon, and thus the material in its club has evolved to withstand repetitive high impacts without undergoing damage or catastrophic failure. So Peshman, could you describe in more detail how in nature the mantis shrimp club is formed?
The club is actually a multi-phase, multi-regional composite. So it combines impact-resistant stiff, hard external region with a softer interior that provides additional dissipation of energy upon impact, that provides greater overall toughness. So the interior region is a three-component structure. And the main component or the bulk part of this material is actually chitin nanofibril, which is organized into highly expanded helicoidal pattern. The second important component is a protein-rich matrix. The main protein in this matrix is quite unique in a sense that it has three-block architectural design, which then result into actually three distinct functionalities that synergistically work together to give the club its unique properties. So it has a domain which is called carbohydrate binding module with the function that it can anchor the overall protein on the surface of chitin nanofibrils. The other part or domain is a silk-like domain which has a reinforcing effect by increasing the interfacial tension between chitin nanofibrils. And finally, it has a mineralizing domain that initiates nucleation and growth of hydroxyapatite minerals on the surface of chitin. And of course, the key to all this is the way that actually mantis shrimp delivers this sophisticated protein matrix into this cholesteric chitin 3D scaffold. So the protein would actually undergo a premolecular self-assembly through this liquid-liquid phase separation process to form these unique protein droplets. So of course, the advantage of the assembly of this protein into this droplet is that they would actually show increased intermolecular interactions and that the low viscosity and low surface tension of these droplets would actually allow greater infiltration and adhesion to chitin scaffold. And also, such a condensated protein droplets would actually increase the local concentration of minerals that are crucially important for the biomineralization step later on.
You wanted to mimic this fine composite structure present in the mantis club. How did you do it?
So we successfully fabricated a biocomposite made of analogous building blocks, namely cellulose as a replacement to chitin, a set of artificial proteins with both cellulose binding modules and acidic domain from mantis shrimp club that regulates the biomineralization and apatite formation, which is both strong and also damage tolerant. So this composite consists of cellulose nanocrystals or CNCs with a long-range helicoidal organization mixed with two types of genetically engineered protein matrices. So one protein was designed to increase the interfacial strength between the chiral pneumatic oriented CNC network through non-covalent interactions. The second protein was designed with the acidic rich domain of the club protein in order to harden, strengthen, and also stiffen the surface of oriented CNCs by mediating nucleation and growth of hydroxyapatite crystals. We process this composite material into intricate shape by manufacturing it into a dental implant crown with this periodic pattern of macro reinforcement and orientation and a biolayer architecture, exactly similar to a human tooth.
These multi-phase nanocomposites are both lightweight and impact resistant, making them perfect also for sport equipment, abrasion-resistant coatings, body armor, electronics, and photonics.
Yeah, first of all, Espoo is the leading innovation hub in the Nordic countries and has been ranked several times as one of the innovation hotspots in the whole European Union. And well, Espoo is hosting a unique ecosystem, thinking about a high density of researchers, high-tech professionals, entrepreneurs, students. It's really a great place to thrive, to do, and to make new things happen. And the Otaniemi campus is a very unique place, and here you find a very high density of high-tech researchers from VTT and Aalto University, but also very close by you find Finnish corporations that make about half of the value in Helsinki Stock Exchange. So we have large anchor companies in this ecosystem. Then we have a very good startup ecosystem that has basically given birth to most of Finland's unicorns. This ecosystem has attracted a little bit over €500 million in venture capital last year. So it's also getting more and more mature, and also for the local companies it's getting easier to raise also larger funding rounds.
If we think about synthetic biology and biotechnology, what kind of infrastructure is available in Espoo?
First of all, Aalto University and VTT, Technical Research Center of Finland, they offer together the infrastructure for bioeconomy companies. And I think this is a very important starting point, that these two big innovation players open up the infrastructure and make it also available for everyone who wants to join in. At the pure numbers, Espoo has the highest education level in Finland, and of course Finland as a country has a very high education level. And then, as Espoo is the place with the highest concentration of international top talent, it's also a place that has been international and also embracing people from all over the world for such a long time.
I think we can safely say today that all of our themes have touched sustainability issues. What are the sustainability goals of the city of Espoo?
Sustainability is a key target for the city of Espoo. Espoo has been ranked as the most sustainable city in Europe a couple of times, and the city of Espoo is very committed to the United Nations Sustainable Development Goals. And last year we have published our voluntary local review on the Sustainable Development Goals. And we try to embrace it in everything that we do, and particularly collaborating with companies, with research organizations, with educational organizations wherever we can to basically find the best solutions for sustainable development. Because for the city of Espoo, it's very clear that we need to do it together.
Thank you so much, Glenn. Moving on from Espoo to look at all of Finland, please welcome Helvi Väisänen, Senior Advisor at Business Finland.
Ladies and gentlemen, hope you and your families are all well in these special times. I am Helvi Väisänen, Senior Advisor at Business Finland. I'm here to tell you about Finland as business environment and encourage you to kickstart your bio-based business in Finland. Our message is simple and clear. Let's get in line with sustainable development. Let's get rid of fossil resources. Let's find a way to generate more wealth, well-being and growth from natural resources. The environmental urgency is stronger than ever. The key drivers are the consumers, the smart investors and the rapidly changing regulation. A whole new generation is coming, and they are not going to accept the way our generation has worked if we do not change. Finland is in the leading position in the bio-based solutions. This is why we can help the rest of the world also to embark on this journey. Business Finland is the government organization for innovation funding, trade, travel and investment promotion. We are headquartered in Helsinki, employ 600 experts in 40 offices globally and in 20 regional offices in Finland. So why work with Finland? When you are looking for an ideal partner, what are you looking for? Well, let me guess. I think if you are a large company, I think you are looking for a business environment with stability, predictability and high level of safety. Perhaps university-industry collaboration in research is of importance to you. And last but not least, I think you are looking for partners that you can trust on, people that do what they promise. And look at this. This is Finland, according to independent studies such as the World Economic Forum, Global Innovation Index, Bloomberg Innovation Index. Finland ranks the best business environment in the world. Number 1 in primary education, the best in the world of innovation, number 1 in university industry research collaboration in the world, the best destination in EU for international business expansion, the most stable country in the world, and for the 4th year in a row, we are the happiest country in the world. We may sit in the darkness for a couple of months in the year, but everything works in this society. People have great work-life balance, and this is why Finland makes so much sense. We offer business opportunities in high-tech and services, in bio and circular economy, clean tech, health, ICT and digitalization, travel and tourism. Finland is the base for international bioproduct manufacturers. You can see from the collection of these logos that most of the big international integrators are here in Finland. And please note, there is room for more companies in our ecosystem. Finland is the epicenter of bio-based growth. Our success is based on a deep understanding of the entire bioproduct value chain, world-class experimental research, integration of cross-disciplinary sciences, and open access pilots and demonstration platforms. So if you are a large company seeking growth and sustainable competitiveness for your company's future, please contact us. We at Invest in Finland are your partner, helping you in opportunity analysis, exploring entry alternatives, data collection and analysis. We facilitate corporate networking with right partners and help you in setting up your business. Our comprehensive services are confidential and complementary. So this is my contact information. We would love to hear from you and set up a time to discuss your goals, understand your challenges, and help you to kickstart your bio-based business in Finland.